Again, welcome everybody to our January keynote webinar. This is actually the 1-year anniversary of our first webinar. And today's webinar is going to be on the management of symptomatic neuromas, a clinical and scientific update. And our speakers will be doctors Kyle Eberlin and William Renthal. So it's my pleasure today to be able to introduce both speakers, starting with Dr. William Renthal, who's an associate professor of neurology at Harvard Medical School. He treats patients with refractory headaches and facial pain. And in addition to treating patients, Dr. Renthal is the director of research at the John Graham Headache Center at Brigham and Women's Hospital. He also does research that's focused on genomic and epigenomic mechanisms that contribute to chronic headaches and pain. And he directs the Harvard Precision Pain Center, which is a collaborative network of pain clinicians and scientists who procure phenotyped human pain-related tissues and characterize those cell types and states using single cell multi-technologies. So thank you for being here today, Dr. Renthal. Our second speaker is Dr. Kyle Eberlin, who is Associate Professor of Surgery at Mass General Hospital, and also the Program Director of Plastic Surgery Residency Program at Harvard as well. Dr. Eberlin's practice focuses on all aspects of hand surgery, peripheral nerve, and microsurgery. He serves on the board of directors for American Society of Plastic Surgeons and the American Association of Hand Surgery. He serves as a section editor for hand and nerve surgery for plastic and reconstructive surgery. He's the section editor for peripheral nerve surgery for plastic and reconstructive surgery. And he's also on the editorial board of hand and orthoplastic surgery. We're very fortunate that Dr. Eberlin is also a member of our education committee, and he has helped us with multiple programs, including moderating a webinar previously and serving as faculty for our in-person courses. So again, thank you both today. And with that, Dr. Eberlin, I will turn it over to you.
Thanks, Steve. And, Will and I are both really excited to be here. Our plan is to make this as interactive as possible. We're going to probably talk for about 45 minutes total between the two of us, and then hopefully have a few moments at the end for some discussion. So we would encourage folks to put questions in the Q&A tab, and we'll try to get to them. I am going to share my screen here. So my task at the beginning is to talk a little bit about where we are clinically with the management of symptomatic neuromas. And I'll share a little bit about how Dr. Renthal and I became partners in this effort and some of the work that we're doing together. For those of you who've heard me talk about this before, there will be a lot of new material actually in some of our recent updates in clinical work at near the end of it. So, and stuff I really haven't spoken about before. So hopefully it's of interest to folks. Talking about neuromas is by far my favorite topic. I think it's really intriguing intellectually and academically as a surgeon when we care for these patients and we're taking out these neuromas. They all are a little different morphologically. They all impact the patients in a different way. And I think we have a long way to go to learn about them. And that's why we're all here. Here are my disclosures. They are relevant to all in the nerve space. But as a brief outline for the next little over 20 minutes, I'll share just some background about neuromas and neuropathic pain. Actually, we'll philosophically talk about what I've learned in caring for these patients. I'll kind of share with the whole audience where we are in 2024 in the surgical management of neuropathic pain, and then we'll highlight some of the clinical work that we're doing research-wise in collaboration with Dr. Renthal and his lab. So, we surgeons and clinicians that take care of these patients see these folks with really, really bad neuropathic pain. And sometimes their pain is due to neuroma, sometimes it's due to other nerve aspects. But when it's due to neuromas, we really don't have a perfect solution surgically. We have things that work, of course, and we'll talk about that. But there's a lot more to it than meets the eye. And that's the one thing I've learned over the last few years is that it's a very multifactorial problem. And we as surgeons and as clinicians tend to ignore this problem a lot. Perhaps not a lot of folks on the call because you're thinking about this, you're here, you want to talk about it and learn about it. But many other providers just really disregard this. And if you don't think about this, you'll never pay attention to and never diagnose it. So the eye only sees what the mind is prepared to comprehend. So we all know that neuromas happen when nerves are injured and they're not connected to their target organ. And every single nerve injury, every single one in the body forms a neuroma. Not all are symptomatic, but every nerve injury forms a neuroma. And when we look at neuromas on the inside, I'm going to play this video. they have this interesting consistency at the terminal aspect being really bulbous very often, and you can see the fascicular architecture proximally.
All right, so these are neuromas of the median and the ulnar nerve. You can see here, this is the terminal aspect of the median neuroma. When we look proximally, you can see fascicular architecture here. But as we cut into it further, there's some evidence of fascicles here. More distally, there's just disorganized tissue, connective tissue, but no axons.
So, from a research standpoint, one of the things that we wondered initially was how do we diagnose symptomatic neuromas? And to really cut to the chase, and this is a paper we wrote, to be diagnosed with a symptomatic neuroma, I think that you have to have neuropathic pain, symptoms in a defined nerve distribution, and a history of nerve injury or suspected nerve injury, and at least one of the items in the bottom blue rectangle, which is a positive Tinel sign, response to local anesthetic injection, or radiographic confirmation. So I think this is an important thing when we're talking about neuromas, when we're talking about the clinical diagnosis. But not all neuropathic pain is secondary to symptomatic neuromas. And I really like this paper that was written by Susan McKinnon and her team and Elspeth Hill, who's now at Yale. It's a really thoughtful way, in my opinion, of categorizing the different types of nerve pain that can occur in the periphery. So neuroma is definitely one category. You can see in the green here, you have a neuroma where you have an injury to the nerve, it forms a neuroma, and that can cause pain. But other nerve problems can cause neuropathic pain as well. Nerve compression, cubital tunnel, carpal tunnel can cause nerve pain. The pain of collateral sprouting, and actually that is really important, I think. And pain for hyperalgesia in the neighboring nerve territories is a very powerful thing, and frankly, I think it's hard to make better. And then the aspect of phantom nerve pain, which is the secondary and tertiary changes that occur in the brain as a result of nerve injury. So I think this framework of understanding the different types of neuropathic pain is critical when we think just about neuromas. And we know that some nerves are more susceptible to developing neuropathic pain. For any of us who deal with injuries in the hand and upper extremity, the radial sensory nerve is a real bad actor. Patients do not do as well when we treat these nerves as they do with other ones. And this is a paper we wrote in PRS a few years ago. But we also know that some patients are more susceptible to neuropathic pain. And as we think about, the scientific aspects of neuroma, this is one of the things that really has driven me and Dr. Renthal and others to kind of try to figure this out is not every patient responds the same way to a nerve injury. Not every patient responds the same way to a nerve compression problem. And, there's probably biologic as well as other factors involved. And we're trying to figure that out. So what I've learned, and this is going to be a lot of saying that I don't know things, which is true. Not all neuropathic pain is due to neuroma, for sure. Not all neuromas are painful. That is definitely true. Not all painful neuromas can be improved with surgery, even if your diagnostic block makes them better. And then individual nerves, as well as individual patients, respond very differently to injury and surgery. And, anecdotally, I've been fortunate to have visiting surgeons from around the world that I kind of work with have come to visit my practice. And it's been very interesting to have different surgeons in different parts of the world say, wow, you see a lot of patients with neuropathic pain that they don't really exist in my country. And I'm not sure that they don't exist, but I think that sometimes there are just bigger fish to fry, so to speak, and people have other problems that they're worried about as opposed to, hey, I have a little numbness in this particular area. So I think it's an interesting dynamic that the interaction between patient and biology. So in 2024, there are many different treatments that we have for the management of symptomatic neuromas and neuropathic pain, and this is both surgical and non-surgical. So this is a paper that we wrote last year that kind of frames what we can do as peripheral nerve surgeons in the management of neuropathic pain, and it's not just the management of neuromas. And again, it really goes into those four categories that Dr. McKinnon and her team described in their paper, which is, we may be able to help with neuromas and nerve compression. Phantom limb pain is a lot more challenging to deal with surgically, even though some of our techniques can make a difference. And then hyperalgesia is, I think, very, very tough. And those of you on the call who take care of a lot of neuropathic pain patients know that those are tough things to make better. So shifting gears and talking just for a moment, this is going to be very brief, about where we are in 2024 with our surgical treatments for neuropathic pain. There are many, many things that we can do to treat neuromas and neuropathic pain. And this paper that I wrote with my good friend Ivan Ducic a few years ago, I think this is now, gosh, it's 6 years ago, but a lot of the themes here have evolved significantly since we wrote this paper. And the idea of giving the nerve a job to do is, I think, become a fundamental tenet of the way we treat neuromas in 2024. And that is to say that we're trying to close the circuit and give the nerve a function and give the nerve a job to do with whatever technique we're using. And it seems like that somehow, and hopefully we can prove this, helps with the central nervous system connections and the whole interface between our peripheral and central nervous system to try to dampen down the painful response to these nerve injuries. And when we think about neuromas, we know that if we have a terminal neuroma where the distal end is not available, that's very different from a neuroma in continuity. And so if we have a neuroma in continuity where the distal end of the nerve is available, we generally want to reconstruct the nerve. And this is a very simple case. I've shown this before, but this is a radial sensory nerve injury where you have neuromas of the two branches of the radial sensory nerve that was untreated. And we have the ability to resect these neuromas and to reconstruct them, in this case with allograft, but with a graft type of reconstruction. And if there's a nerve injury where you have the terminal end available, it is my recommendation to reconstruct the nerve in the vast majority of cases. Now, we studied this when we looked at digital neuromas. We looked at patients with not a huge number of patients, but a certain number of patients who had digital neuromas that were treated surgically, either reconstructed or had, quote, ablative techniques. And we followed them over a long term. This was more than 7 years of follow-up. And what we found was that, and this is somewhat intuitive, I think, but if you reconstruct the nerve after excising the digital neuroma, patients do better than just kind of ablating the nerve and resecting the neuroma and burying it proximally. So this makes sense. I think that, we want to recapitulate whatever the original function was, but this is something that we relatively showed in this paper. So if we have an end neuroma, that's when we get into all these techniques, which everyone on the call is familiar with. I'm not going to go through what they are. And TMR and RPNI have kind of led the charge, so to speak, for the management of end neuromas. And these are really important techniques that were developed by Greg Dumanian and Paul Sedurna. They have changed the game for amputees and for the way we treat these nerves, but it doesn't have to be a competition. And I would say that, I use both of them. I tend to be more of a TMR person than an RPNI person, but, I think gone are the days where it's just people up on stage arguing about which one of these two are better. That's not really the point anymore. And we're trying to use our paradigms to address these nerves in the best way. And the reason that these techniques seem to work is they both provide open end plates for the regenerating nerve. So as the nerve regenerates, it's providing and conferring a functional destination for these nerves. And that seems to be why they work pretty well. For anyone that's interested in TMR or is doing TMR, I would direct you to this paper that I wrote with quite a number of other authors last year, where many of us who do a lot of this surgery got together and tried to come up with a consensus approach to how this is performed. And this is an open access paper, as you can see, but there's a lot of information here about how many nerves do you address, when should you do TMR, where do you do your coaptations, proximal or distal, and then what do you do with the donor nerve? So if anyone's interested, please feel free to read this. I do think it would be useful if you're doing TMR in your practice. But like I said, it's not only about TMR and RPNI. We do not know which surgical techniques are best for which patients. And as we talk about the science in a few moments, what we're really going to be focusing on is, well, what else can we do? I suspect and I hope that Will and I, in a number of years, will have some therapeutic targets or things that we can do with nerve endings to make them less painful. And there are other neuroma techniques available. And for instance, Sami Tufaha and Ellen Belzberg and the team at Hopkins came up with this idea of vascularized denervated muscle targets, which is somewhat the same as TMR and RPNI. It's the same theme, but a different way to do it. And I think there's a lot of promise there. And one of the things that my partner Ian Valerio and I do all the time is to combine TMR and RPNI. And this is a paper that we wrote up about the technique of how we do this. It's not rocket science. We do an end-to-end nerve transfer as you would do for, quote, normal TMR. And then we wrap the coaptation in denervated but vascularized muscle nearby. So if there's any escaping axons, they're captured by this muscle. There are other things that we can do also. And Rob Hagan and I came up with this concept of a reset neurectomy, where you basically perform a neurotomy or even a neurectomy proximal to the area where the nerve is painful or injured. And essentially, you're doing a Control-Alt-Delete of the nerve itself and sending the axons back downstream. I do think there are limited indications for this, but it's just another way that you could treat a painful nerve with the reset neurectomy. And then finally, there's tons of other technology that is either out or coming out that is going to help us, I think, as surgeons and as clinicians to take care of these problems. And, one of the things that I do occasionally is relocation nerve grafting, where when you have an injury and you have neuromas and you're going to resect them, these are, of course, digital neuromas, and you're trying to get the nerve endings and the painful nerve away from the working surface of the hand where the person's going to be grasping things. By resecting the neuromas, using an allograft to direct the regenerating neurons toward the dorsal aspect of the hand away from the working surface. This is called relocation nerve grafting. So I would say in total, and I went through all that stuff relatively briefly because I want to make sure that we have time for discussion, but also to talk about our scientific efforts. We've come a long way with our clinical management and surgical management of the symptomatic neuroma, but we have a long way to go. And this is what makes it so exciting for me, and why I'm interested in this topic is, this is not like a distal radius fracture where we have lots of solutions and we have a pretty good solution for the problem. So Will's going to talk a little more about this, but I want to highlight the fact that, I feel very lucky to be in Boston with a lot of really, really smart scientific folks. And, Dr. Renthal, from whom you will hear, as well as Dr. Wolf and Dr. Wenger, really are world experts in the management of pain and the scientific research into pain. And it was very fortuitous how our collaboration developed. But it's really been a great part of my practice, and also in looking forward to how we can move things forward in the future. So we've started this Harvard Precision Pain Center that Will runs, and he'll talk a little more about it. And from a clinical standpoint, we really have a great team. Ian Valerio, my partner, and Neil Chen, who's an orthopedic surgeon with whom I work, and all of our researchers, and I don't have all their pictures up here, but Flores, who's here in the center, he's kind of the center point of our research team and has done incredible work, and I'm very grateful to work with him. But, other folks on here, Mike, Rachel, Karen, Andy, and Chuck, and Carla, and Daniel, and a few others on our team that really are the driving force behind our clinical research. So we have collected over more than 100 neuromas. I don't know what the exact number is now, from patients, and I'm going to share with you kind of how we do this in a practical sense in our clinical practice. So before the operation, obviously the patient's going to undergo some sort of neuroma excision, regardless of what we're doing with the nerve afterwards. And we've excluded patients who are under 18 and who have malignancy, even though there are a lot of patients with malignancy who undergo, or prior malignancy who undergo these types of surgeries. So the first thing that we do is we do a preoperative interview. We have a ton of pain scales, largely dictated to us by the NIH. We are using pain sketches now for them to draw out where their pain is. If they're an amputee, they can draw on the residual limb as well as in the phantom part of their limb where the pain is. And we collect all of that information preoperatively. In the operating room, we, of course, are excising the neuromas surgically. We have part of our research team is there. They're collecting the samples. They do a fair amount of work on the back table to kind of remove any of the connective tissue around it to make sure that the neuromas themselves are visible, but that we're really just looking at the neuromas, and you'll see why in a moment. And then they go on dry ice and they go into a deep freezer. And we do have some fresh samples that we've studied, and Will can speak to it, but largely speaking, this is how we collect it. And then I'm not going to talk about this, but a lot of the analytic part then goes to Dr. Renthal and his team. And I know they're kind of in the early phases of the analysis, but I think we're going to hopefully have some really, really interesting data, which combines the demographic information for the patients. We know how painful each neuroma is. You can imagine for a below-knee amputee, we will know which nerve has which pain score, and we can hopefully correlate that with the biology. I think that's very, very exciting looking forward. So some of the research questions that we had really clinically, any of you have taken out neuromas know that they come in many different shapes and sizes. So one of the questions was, do they come in different morphologies? And the answer is yes, they do. So looking at some of our early samples, you can see here for this particular study, the average time for these 45 neuromas from the time of the nerve injury to the time of neuroma excision was 7 years. you can see what type of amputation the patients have, but and that there's a mixture of upper and lower extremity specimens. But when you look at all these different neuromas and you look around the screen here, there are different sizes nerves. They have different shapes of how bulbous they are potentially, or even just what the morphology is. So we came up with this classification system that was not reported previously in the literature. We've actually submitted this paper. Hopefully it gets accepted somewhere into a reasonable journal. But where we basically categorize them into bulbous neuromas, fusiform neuromas, and atypical neuromas. And we said to ourselves, gosh, are there any differences between the pain the pain phenotype for these different neuromorphologies? So interestingly, when you look at these different types of neuromas, the atypical nerves tend to be more painful. I can't tell you why that is yet, but that is something that we noticed in this study and also in some of the subsequent patients that we've analyzed, that the atypical neuromas tend to have higher pain scores ahead of surgery. We also noticed that atypical neuromas, those patients tend to have a higher prevalence of diabetes, as well as a higher prevalence of peripheral vascular disease. So there may be some other patient factors that are related to this. There may be some local factors in the tissues that contribute to why the neuromas are shaped the way they are. And we don't really understand this completely yet, but it is something that we have noticed that, that atypical neuromas tend to be a bit more painful. When we look at these neuromas, some of them have distal neurite outgrowth. So you'll look at the nerve, you'll be looking at the neuroma, and you will see that there has been outgrowth of the neuroma, the axons past the neuroma bulb. And it's pretty interesting when we see that. And it tends to be more common with the atypical neuromas. So perhaps there's something biologically with these neuromas that are related to it. So a couple other questions. I'm going to wrap up very soon. Do neuromas continue to grow over time? Does the size of the neuroma impact the degree of pain? So we've looked at both of these questions. So we wanted to come up with a way to measure the diameter of the neuroma compared to the diameter of the native nerve. And basically what we do is we measure the widest part of the neuroma, and then we measure the native nerve 5 millimeters proximal to where the neuroma begins. And we've coined this the neuroma to nerve ratio. So basically trying to decide how big is the neuroma related to how big to the size of the nerve. And it turns out that, and I'm just going to show you, cut to the chase here. If you look at the neuroma to nerve ratio, on average, for all the neuromas we've analyzed, it's about 2.5, meaning that most neuromas, or on average, the neuroma itself is 2.5 times the width or the diameter of the native nerve itself. Now, this in and of itself is not terribly interesting, but we looked a little bit more into the details. So over time, I've always wondered whether neuromas continue to grow over time or whether they reach some plateau and then they just sort of remain quiescent in the body. And when we look, and you can look at the x-axis of this graph, we have some patients who've had neuromas excised many, many years afterwards. It does seem like neuromas grow over time, and this is statistically significant. So it is my belief that once there's a neuroma, it does continue to have trophic factors and change over time based on this data. When we look at pain and look at just the ratio, the neuroma to nerve ratio in pain, you might think, well, larger neuromas, relatively speaking, would have more pain. And there is a trend toward this. The p-value is 0.08, so it's not statistically significant, so I can't say that it is correlated, but there is a bit of a trend in that direction, but nothing confirmatory just yet. When we look at the individual nerves, this is where I think it gets a little interesting morphologically. When we look at the different types of nerves and how large their neuroma to nerve ratio is, you can see that some of the largest ones that we find are the saphenous nerve, the sural nerve, and some of the sensory nerves. The smaller sensory nerves tend, for whatever reason, to have a larger neuroma to nerve ratio. And you can see that here. And this is also, you can see it in this graph, where the smaller the diameter of the nerve, the native nerve, the larger, relatively speaking, the neuroma is, and the larger the neuroma to nerve ratio is. So that's pretty interesting, I think. And I think another question that comes up in this topic is, what about the difference between mixed nerves, or motor nerves, and sensory nerves? And we found that sensory nerves develop larger neuromas than do mixed nerves. And that probably has some biologic implications, I would imagine. But it doesn't, we haven't shown yet that there's a difference in pain. And finally, just to wrap things up, we're starting to study, some of the histology and staining of neuromas for both sensory and motor components. Vlad Terashenko, who is working with our team, is spearheading a lot of this work as well. Has done a great job with it. And then finally, I want to say this is to just mention that we've noticed this as well that I'm really interested in. So we have noticed, I'm just going to show you this X-ray of a below-knee amputee where you have this large fibular spike of heterotopic ossification. Now, I and our team have noticed over taking care of many amputees, that there is always a neuroma at the end of that heterotopic ossification. I can predict that right at the end of that nerve, at the distal end of the fibula, right at the end of the bone, at the distal end of the fibula, there's going to be a neuroma there. And there has to be some biologic relationship there. And I believe that it's usually that I think the growth factors for neuroma development are feeding into the heterotopic ossification formation. And we've studied this, we submitted this paper, and I'm just going to say that when patients have fibular heterotopic ossification, they tend to have more symptomatic neuromas in the perineal nerve distribution. And when they have tibial heterotopic ossification, it seems to be more in the tibial nerve distribution. So this is interesting, and it's hard to separate pain from HO versus pain from neuromas, but I do think that there's biologic overlap here. I really, really believe that, and it's our job to prove it. So I'm going to finish by saying that, and I think Will may touch on this a little bit, that we recently had a paper published in Pain that kind of summarizes our understanding of the biology of symptomatic neuromas that I would encourage you to read if you're interested. And I think the future is that we're going to hopefully develop some targeted therapies, and as we understand the biology better, we'll be able to better treat these patients. So I'm going to stop there. I stated 25 minutes, and I think I hope this was of interest to folks, and I'm really excited to hear what Will has to say. And again, we'd encourage you to ask questions in the chat, and appreciate everyone being here. So I'm going to turn it over to Will.
Perfect. All right. Well, it's a real pleasure to be here, and this collaboration that I've gotten to have with Dr. Eberlin and his team has just been really phenomenal. And broadly speaking, I'd like to talk a little bit more about some of the cell types and states of human neuromas. That's the ultimate goal of this work, is to understand kind of the symphony of communication between the different cell types in these structures, and also get some ideas for new potential therapeutic targets. These are my disclosures. None of them are relevant for the talk today. And so the overview of the presentation is going to, again, just be about the cell types and states that are involved in nerve injury and repair, our overall approach to studying human neuromas, and then some early results and future directions that we're taking now. As Kyle just mentioned, a lot of the summary of some of the mechanisms I'll be talking about today are written in this beautiful paper that just came out. So if you need any more depth or have any questions we don't have time for, just please feel free to turn to that. So I'm sure you all are experts on, there's a massive structural difference between sciatic neuromas or any neuroma and the normal nerve. Perhaps the most obvious that you can see here is that there's a disordered structure in the neuroma compared to the normal nerve, and that the myelination patterns are quite different, much thicker myelination in normal nerve than in neuromas. There's also many different, when you zoom in, many different other cell types that are present in traumatic neuromas, especially marked by fibrosis that occurs and immune infiltrates as well. And so our goal is really to try to leverage some of the new technologies that have come out over the past few years to try to understand those different cell types in human neuromas. And so the general process that's thought to occur in neuroma formation, and actually any type of regeneration after nerve injury, is that there's a signal that gets released during the acute injury when the nerve is injured, often involves macrophages, local resident macrophages, or other immune cell types that release growth factors into the blood and nearby tissue. That activates certain Schwann cell subtypes and recruits other regenerative macrophages and immune cell types that then help grow this nerve back into the target tissue. And this is what you would get with a successful nerve regeneration, which obviously does not occur in the case of a neuroma, where these factors either don't get activated properly or get pathologically activated, or there's too large of a gap between or basically the tube gets damaged in such a way so the nerve can't extend and goes into the wrong direction, or the signals of the target tissue for some reason aren't getting to the trophic factors, aren't getting to the nerve, and neuroma forms. So the biology that has been described in this has been actually quite well established over the past 30, 40 years in animal models. And I'll take you through a little bit of that early data now, and then kind of how we're trying to unite this with understanding of what's going on in patients. I'm fundamentally a molecular cellular biologist and want to understand what cell types and states of those cells are driving pathology. And so as we know, and I'll talk a little bit more, cells like macrophages are very different in different places. And so it's really important to understand them in the context of the disease you're studying. And so this is kind of the group of the players, the key players in terms of nerve repair and neuroma formation. These are macrophages and other immune cell types, fibroblasts, the endothelial cells and pericytes that line the blood vessels that are that feed these nerves, the Schwann cells and other glial cells. And then ultimately the neuron, which we think is probably the key reason why all these other cells exist, is to try to get that neuron to innervate the tissue that it was supposed to innervate initially. And so let's just go through a little bit about what's been described with these different cell types. The Schwann cell was initially, this is a group of nerves that is just plated in a cell culture dish, and you can see that they form little, outgrowths, neurites that occur kind of symmetrically. And it was described back in the early 90s that when you put a Schwann cell in the same culture, these little processes will grow towards the Schwann cell. And so this was kind of the initial idea that there are growth factors these Schwann cells are secreting that cause neurons to grow in a specific fashion. But so this has now been developed over the last few decades and described in more depth. And one of the key factors that's now understood is that these Schwann cells get a signal upon injury to actually transform their cell state, their cell identity, into what's called a repair Schwann cell at the site of injury. And these factors, this transformation requires the upregulation of a transcription factor called c-Jun. And this allows these Schwann cells to release factors that are critical for nerve regeneration and that kind of directional outgrowth of neurites that I just showed you. And just some data to kind of highlight the differences when you can't make that transcriptional switch or that cell state switch in Schwann cells. The regeneration is much less in the knockout mouse compared to a wild type. And so this is pretty clear evidence describing the importance of both the cell type and cell state in that tissue for normal nerve regeneration. What guides the Schwann cells, though, there's a massive infiltration into the injury site of fibroblasts as well. And so this is a red stain here for fibroblasts compared in the injured nerve compared to the control nerve here. And these fibroblasts are known to release certain factors, one of which is called Eberlin, which is a neurotrophic signal that can guide both Schwann cells and neurons to grow in a structured fashion, kind of in a straight linear fashion you see here in the normal mouse, whereas in the knockout mouse for Eberlin, excuse me, there's much more disordered neuron outgrowth. So this is another important cell type, and fibroblasts are known to be very dynamic cells as well. And clearly, we need to get better understanding of how these cells are regulated in neuromas themselves. Very recently, actually, as of just this past week, what's guiding the fibroblasts and the Schwann cells, it's actually thought now, as of a few weeks ago, to be the blood vessels, the structure of these blood vessels that are lining the injured crushed site of the nerve. If these are still intact to some extent, it's thought that they can serve as a scaffold for fibroblasts and other and Schwann cells to kind of guide that nerve through to repair properly. And this is all, this has long been thought, but the evidence for it's been really difficult to come by. And this really clever study took a mouse where the vessels only, which by making a mutant mouse of this plexin, the vessels themselves are disordered after injury. And so whereas the nerves and Schwann cells and all the other cell types are roughly normal. And they found that you get this kind of disordered scarring that occurs that's very similar to a neuroma in these animals just by disrupting the structure of these blood vessels. Because these videos are so pretty, I just want to highlight what it looks like. Green here is kind of paradoxically the endothelial cells, and the red is the perineural tube. And you can see in a normal animal, you get nice linear perineural tubes that follow the vessels. But in the mutant, which you can see here, the blood vessels are all disordered. In that mouse, you get very disordered perineural tubes, especially after regeneration. So, or after nerve injury. So I think that this is, kind of talking about the symphony of cell types that are having to interact with each other to make successful nerve regeneration. And when that's disordered, you end up getting symptomatic neuromas. We can't talk about neuromas without the nerves themselves. So when injury occurs, you have an intrinsic neural regeneration response. This allows the nerve to regrow in response to all of those factors and along the fibers that we just talked about, or the vessels we just talked about. And in order for that to occur, the neurons themselves upregulate a transcription factor, one of which is ATF3. This is the ATF3 staining in the injured DRG neurons. This is in the DRG, but this is along the whole axon. And this process, when you overexpress this ATF3 protein, you get much longer, much faster regeneration than you do in a control animal. And if you delete this factor and prevent it from being upregulated after injury, you can see that the repair of this nerve, the functional recovery, basically the sensitivity of the paw in a mouse, in the knockout mice, is much longer and much more incomplete than the normal recovery, which usually occurs within a couple of weeks after injury. So, this shows that all of these cell types are really critical, functioning together to get successful regeneration. We've now mapped out at single cell resolution how these neurons respond, and also other immune cells and glial cells along this nerve across a variety of injury responses, including nerve transection, nerve crush, and spinal nerve transection, sciatic nerve transection, various chemotherapy-induced neuropathies, inflammatory pains signals. And you can go look up how every cell type in this structure in the DRG changes in response to these different stimuli. And there's really some dramatic changes that I think are really interesting, and our lab is still very excited about studying in more depth. And then I just want to use this last cell type, the macrophage, to kind of highlight how important the tissue that you're studying is to the function of a cell. Because when you put macrophages inside of a nerve, they act as kind of neurotrophic factors and make the nerve grow towards the macrophage. However, when you delete them from the systemic, from the whole system, you actually get less regeneration. And so I think that there's this kind of paradoxical response between macrophages that is really important for understanding in this site of interest. And so just to highlight that the goal that we're trying to do is map out all these different interactions between these different cell types, which has been done to some extent in mice, but really not in humans. And what we found, especially in the pain field, is the importance of studying human cells, because there were really important differences in channel expression in therapeutic targets that could potentially explain why certain things haven't translated. And so our goal is to really map these out and understand what can be modeled well in lower species versus what can't be and really needs human models. And so this has been the kind of genesis of this Harvard Precision Pain Center, which I've been really fortunate to work with Dr. Eberlin and Dr. Wolf and these other really fantastic PIs, where we're trying to build this neuroglial immune axis or atlas, where we can describe all of the factors that are regulated and expressed in sensory ganglia and neurons and glial cells here, as well as those in the neuromas, and kind of build a connector between how these cells are all communicating to each other, using cutting-edge single-cell biology tools that I think are really changing the way we're understanding how cells communicate. And just to give you a kind of a background of what this is, we were taking either ganglia from postmortem donors or neuromas from the neuroma bank that Kyle has just talked about, and we dissociate these cells into single-cell suspensions. We then have microfluidics devices that can encapsulate into single droplets an individual cell and a barcode that we include into that droplet. And this allows us to label all of the RNA and all of the molecules in that cell individually. And that once we sequence them, we can now we know which cells they come from, and we can map out for each cell the number of counts of each mRNA, for instance, for every gene in the genome, and for every cell that we sequence. And so we can build these massive matrices of millions of cells, and how each cell is expressing different genes. There's obviously a huge amount of data, and requires kind of informatic tools for humans to understand. And so what we do is we use graph clustering or dimensionality reduction tools that allow us to cluster cells into different cell types or cell states. And so cells, these types of plots I'll show, cells that cluster together are more similar in their transcriptomes versus in those that are separate are more different. And so we've built atlases now of the human DRG and trigeminal ganglia. And just to kind of show you what we're across multiple species, model organisms, as well as human donors. And these are the neuronal cell types. We've done this for non-neuronal cell types as well, but these are just the neurons here, just so you can see what these plots look like, and the number of distinct neuronal subtypes that exist within human and model organisms. It turns out that the broad cell types are actually quite similar across species, so that's actually really fortunate. I think in give some credence to modeling neurodegeneration in animal models. But obviously there are a lot of differences between human and mice too, so that's kind of where the hard work comes in. You can go through and we're building these web resources so that anybody can go into these websites and look up which genes they're interested in and what cell types they're expressed in, or what cell types they're interested in, and find out all of the transcripts that are either expressed in that cell type or changed after different injury models across multiple species. And so this is kind of now getting back to the neuroma side, which as Dr. Eberlin just mentioned, we're in the beginning stages of really starting to characterize. His group has done a fantastic job, phenotyping these patients and collecting neuromas from them. And then, where our group comes in is in taking these samples and then performing multi-omic analyses to try to understand what's going on at a single cell resolution. And a lot of this work is done with a large group of investigators, many of whom are led by Dr. Clifford Wolf's team. And my lab is really driving force behind trying to understand the analytics and bring some of these multi-omic tools to these data sets. So the things we're characterizing are human genetics. So we genotype all of the samples. We perform single cell RNA sequencing on all of the samples. We perform spatial transcriptomics, soon to be on all of the samples, and then proteomics. And so we're just in the very early stages of this, so I don't have a lot of data to show, but we're hoping that this kind of multi-omic interpretation can then, or analysis of every sample, can then be correlated with human pain scores, so we can understand which of the cell types, cell states, and features are actually driving or correlating with the symptomatic neuroma, and it gives us some more targeted ways to think about treating them. The tissue bank that Dr. Eberlin's group has created has at least 95 neuromas at this point, and 106 control nerves. The way we've been processing these samples is to dissect the neuroma separately from the proximal nerve of the same individual, and then also separately a control nerve from a different individual, and perform these multi-omic analyses, and ultimately correlating with pain severity. This is some early data from a few neuromas. I think this is four neuromas and 4 control nerves, where we perform single cell sequencing and of each of these samples and look to see which cell types are present in each of these. And so these are the cell types that we've found so far in these human neuromas. And I'm just going to now, instead of coloring each of these dots based on the cell type, I'm going to color them based on whether they're neuromas or from the proximal nerve. And what you can see here is that there's largely similar cell types between the neuroma and the proximal nerve of the same kind of injured nerve. But two cell types that really stand out as different are the adipocytes, which are adipocyte-like cells. We actually haven't formally proven they're adipocytes, but they look transcriptionally a lot like adipocytes, which are really only present in the neuroma samples, and macrophages, which look like they're much more present in the neuroma than in the proximal nerve. So we're pretty excited about some of these early findings. There are also a lot of transcriptional differences in each of these other subtypes, especially these fibroblasts that we're really excited about, and also we just generate a lot more data to see how stable these are over a diversity of individuals. We've also, as I mentioned, been performing proteomics of these samples, and this is just we've done a lot more samples for proteomics, and this is a lot of data here. There's upwards of 10 to 20,000 proteins per individual, usually around 10,000 we measure, and we can. It's a lot of data, much like we just described. So we're using principal component analysis here to just kind of simplify big picture changes between neuroma cases and controls, or in the proximal control. And you can see there's actually some differences here in some individuals that look very dramatic between the neuroma and control nerves. So, we're digging in now to try to understand what this variability is, and what proteins and phosphoproteins are actually driving these big differences between neuromas and controls in terms of proteomics. And finally, this is a recent study that was just posted on medRxiv, where they've correlated, a couple of the expression of a couple of genes with pain scores of neuromas. And I think this is the type of work that we're really excited about doing, except kind of genome wide. And I'm really excited about trying to identify some of these features that may predict pain score as well or even better than what's reported here. And so with that, I just wanted to thank, obviously, the patients who are joining these studies, without whom none of this would really be possible. The whole Harvard Precision Center, it's just been fantastic to work with everybody on both the clinical expertise, the basic science expertise, and bringing everybody together has been just a thrill of a lifetime. And then obviously the support from NIH that's really funding the center, and all obviously the foundations that are coming together to support this work too. And just want to highlight that Dr. Eberlin was on the Boston Globe for some of this work. So if you haven't seen this article, please feel free to join and read it. And I think now we'll take questions.
Thanks, Will. That was fantastic, and I think at a really high level, hopefully, what everyone that's listening can see, or anyone that's listening in the future can see, is that I think to make a difference for these patients, we're going to have to combine clinical and scientific expertise to really get to the root cause and to figure out, biologically what we can do. And so that's, I think, the point of our collaboration. There have been a few questions in the chat. They're kind of clinical ones. I would encourage anyone else that has a question, please put it in there now. We have about 7 minutes left to go through things. But the first question was, do you see more neuromas like a chain on a single nerve, like saphenous after injury? So I think the question is asking whether you see a neuroma just at the end or if you see neuromas along the course of the nerve. Generally speaking, neuromas occur at the end of the nerve, but I have seen numerous instances where there are like beads on a string of an affected nerve, and I have seen that predominantly in the case of a traction injury to the nerve. So you can imagine, historically, when people did amputations and they did a traction neurectomy and pulled the nerve like a tug of war, we have had situations where we've seen neuromas up the entire course of the nerve, and it's really remarkable when you see it. So generally speaking, that they're at the end, but they can be if you have a traction injury along the course. Second question is about there was a patient question about, looks like a patient that the surgeon has performed these nerve techniques on and still is having pain. They're really challenging, and there's really, in my opinion, not necessarily always a surgical solution for these patients. And the things that are worth thinking about now in terms of delivering care to these patients, if they've, quote, failed or have not succeeded with some of these sophisticated nerve techniques, is sometimes stimulation, either peripheral nerve stimulation or spinal cord stimulation, is something worth considering. And it's not a panacea, it's not magic, but those are the things that we're thinking about clinically for those patients. Now, Will, I'm sure you have thoughts about these kind of really tough pain patients who failed all surgeries. What do you do with them when you see them?
Yeah, I mean, I think at that point, it's really, medication management and physical therapy and coping strategies. Unfortunately, all we have right now. And I think, because of how challenging that can be for people. It really is, I think that's the key motivation for this work, to try to identify better targets.
Some more questions are coming in. The next one, and I'll try to get through them, the rest of them that are here in the next 5 minutes, but question about the best technique for MRI detection of neuromas. There's an enormous interest in imaging of neuromas. And I would say that, Daryl Sneeg, who's very involved in the Global Nerve Foundation, who's a radiologist in special surgery in New York, really has led the charge around the country and world for MRI imaging. And his technique of MR neurography, which has been adopted by many centers, I think is really kind of the way to go. And he's given a webinar, and Steve, you can chime in. I'm sure it's recorded, and I'm sure that if you're interested, I would recommend listening to Daryl Sneeg. And if Daryl is ever watching this, every time Daryl goes to a meeting, I was just telling him this recently, every surgeon in the room is trying to recruit him to come to their institution. It's sort of he's like a celebrity athlete that, every owner is trying to get him to come. It's pretty funny, but he's really kind of led the charge there. Next question. Somebody asked me specifically, do you compare post-op neuropathic pain and below-knee amputations with and without fibula excision? Shouldn't the excision of the fibula prevent HO at the tip of the fibula and therefore neuroma pain? Well, almost every time that someone develops heterotopic ossification, they've, of course, had their fibula and tibia both cut. But it's interesting when we see just the fibula develop HO and not the tibia. And I believe that it's largely nerve growth factor driven. And so what we're studying now is we're trying to determine if the treatment of these nerve endings with TMR or RPNI at the time of amputation prevents the development of heterotopic ossification. I think it might. I don't know that for sure, but it's possible. And so we're going to try to figure that out. The next question was how neuromas can be identified. What significant tests for this? Well, I mentioned the criteria that we use from a clinical diagnostic standpoint, but Will, when you're dealing with patients with head and neck problems, and let's say they've had prior surgery or spine intervention or even neurosurgery, how do you think about that, and how do you figure out if they have a neuroma?
Yeah, I mean, we don't typically differentiate whether there's a neuroma or neuropathic pain, kind of not otherwise specified in terms of the clinical management, unless there's a traumatic injury or a tumor. And I think that's just because the management is not different yet. I think that it should be, but it isn't. So we usually just treat them as neuropathic pain.
A question for you, Will. It says the macrophages profiled in the single cell analysis of neuromas is interesting. Do you think that if you could block macrophages from getting into the nerve, you could block neuroma development?
Well, I think this is a great question. Kind of, you take a step back philosophically, the question is, how can we coordinate cell-cell interactions in the neuroma to take advantage of what they're normally doing in both physiological and pathological states. So macrophages are one of them, but as I mentioned, like macrophages can be complicated. They can be helpful and harmful. And so I'm thinking, of ways to take, in this kind of era of cell therapy, ways to take patients' own cells, engineer them to do what we want them to do. So macrophages, and only, let's say, take macrophages that are doing, let's say, analgesic effects. And we can potentially one day deliver these, once we understand the signaling processes well enough, directly to sites of neuroma and maybe offer them, these patients, significant relief. But this is still early days. I think cell therapies for these types of disorders, they're really reserved for cancer treatment at the moment, but this is in the kind of on the horizon.
That's great. Well, maybe we'll go through one more question before we wrap up. It looks like there's some questions about the types of medications that are often recommended for these patients. I'll say, in my very small brain surgeon perspective, we often use gabapentin or Lyrica as a first-line treatment. I found that something like amitriptyline, one of the tricyclics, can be helpful if patients are having trouble sleeping, and it's it's a QHS or once a day at nighttime dosing. So those are often what we use up front. Will, what would you add to that?
Yeah, I'd say those are our first line go-to for neuropathic pain as well. For all the same reasons, yeah.
That's good. Final question I'll answer, just because this comes up a lot when talking about TMR. What happens when a sensory nerve is sutured to a motor nerve branch, such as TMR and digital neuromas? Well, nobody really knows the answer to that question. It does seem like taking a sensory nerve that has a neuroma, removing the neuroma, and doing a TMR into an adjacent motor nerve does seem to provide benefit. And there's probably a combination of pruning that happens, as well as the fact that, even in sensory nerves, or quote, pure sensory nerves, there are autonomic motor fibers in there. So it's probably a combination of the pruning that happens, as well as the different sort of cell types, if you will, within the nerve that are contributing. But it's something that we need to better understand moving forward. But it does seem to work clinically. But I guess the clinical part's ahead of the scientific understanding. So I think with that, I think we'll wrap up. I don't know if Steve, you want to say any final words, but we really appreciate everyone's time. We hope that this was of interest to everyone. And I'll just say to Will, I'm so proud and happy to have the collaboration with you and your team, and we look forward to future developments.
Excellent. Thank you both. I really appreciate the time and the great presentations. Thank you, everybody, for the interaction and questions. We will send out a follow-up email, and we will we'll put in the link for Dr. Sneeg's webinar with Dr. Tan as well. So if there's any further questions, hopefully that will help. But thank you all. And again, please help me thanking the speakers, and we will follow up afterwards. So thank you.
Have a great day, everyone.
Yeah.
Great night. See ya.
Bye.